Igor for an extremely generous and kind introduction, and Neil for being a terrific host, and it's just been a wonderful couple days, and my only regrets is that I didn't save this until about February when I'd probably really be needing it now. And again, as per said, I feel like I've got so many friends and colleagues here for decades, and I really always feel sort of like I'm coming to a second home when I come back to UCSD. So thank you guys so much for just being terrific hosts and a great visit. So I'm going to talk today about our work over the last 15 or 18 years on post-traumatic stress, on fear, on the circuits, try to stay true to the name of this wonderful new building, on behavioral circuits. And I want to talk a little bit about why this has been the area that I've chosen to focus most of my career on. And I think it's because, as so many of us, we want to make a difference in the area of mental health. And for me, trained as a molecular biologist and then did fellowship with Mike Davis, really wanted to do this from sort of a bottom-up circuitry approach. And I have been struck by how particularly amygdala-based functioning as a reductionistic yet still translationally relevant approach makes sense. And so I think PTSD, amongst the complexity of so many of our disorders, is one that remains retractable in that, as I'll talk about, we have a shared fear neurocircuitry across models. We know when PTSD starts. It starts at the time of the trauma. We can certainly talk more about the potential caveats of that. But we could argue from that it's tractable in a temporal way, in a way that most psychiatric disorders are not. And that both the learning of the trauma, the association of the initial trauma experience, as well as the learning and plasticity involved in recovering from trauma, very much engage learning and memory processes. And these are areas that have been some of the most, where some of the most progress has been made over the last several decades in the area of neuroscience. So our goal then is to have a molecular neurobiology of PTSD that incorporates biomarkers leading to new interventions and treatments. And just remind everybody of the circuits at work here. So I'm going to primarily be talking about the amygdala, really as the final common pathway of output, but this is in purpose of reminding you that I don't think the amygdala is the whole thing by any means. It's part of really an emotion regulation circuit. Everything from medial prefrontal that's most consistently acting in a way that's opposing the amygdala and inhibiting amygdala output. Dorsal anterior cingulate that seems to be fear processing, may be akin to the prelimbic in rodents and seems to be enhancing amygdala fear and emotion output. And then the hippocampus, which is really modulating amygdala in complex ways, both in terms of contextualization as well as the context required for learning to inhibit or extinguish memories. And I'll be coming back to that throughout. This is a recent review by Kay Tai and Patricia Janik, and just really nicely lays out the fact that the amygdala, or a function very much related to the amygdala, has really been conserved throughout vertebrate species, and certainly across all mammals it's very similar in its both circuits, in its connectivity, and its function. And this slide from Joe Ledoux reminds us of why the amygdala is so important in a evolutionary sense. So that in a very rapid way, we're able to, in a pre-conscious, really in hundreds of milliseconds way, activate anything that seems to be a threat. So I like this picture especially because I was talking to Jennifer on the way in this morning. We were talking about her fear of sharks and how unfortunate that is in San Diego. And my wife, we have lived in Atlanta for a long time, and there's a lot of snakes in the South. And so we'll go walking on a nice walk in the evening, and we'll be enjoying ourselves, having a nice talk, listening to the birds. And then before I know it, Betsy's jumping up and down screaming, oh my God, she's about 10 feet back. And she, sorry, that was just a stick. And they're always sticks. But it doesn't matter because her amygdala knows and is going to keep her from getting bit by a snake no matter what. But the problem with this system is that in our modern day world, we're much more likely to be activating this system through the traumas around us than we are things that are true threats. And with post-traumatic stress disorder, this system seems to be one in which this evolutionary conservation to have one trial immediate learning that changes your behavior forever out can lead to all sorts of problems related to anxiety, hypervigilance, memory alterations, flashbacks, intrusive memories. And particularly with PTSD, it's almost as if people live in an emotional black hole where everything becomes associated with this generalized fear that brings them back to those triggers and traumas. So PTSD is certainly well known in our military cohorts, and I'm preaching to the converted here. So much of the great work in the countries come out of UCSD, everything from STARS to Marine Resilience to Intrust through the VA programs, and really the UCSD's terrific ability to work with the real critical mass of military that's located here. A lot of our work, in addition to collaborating with those sorts of programs and some of the Atlanta VA, has really come from inner city Atlanta. And I'll talk more about this later, but we've interviewed over the last 15 years almost 10,000 people from inner city Atlanta who are attending Grady Memorial Hospital, one of the largest remaining charity hospitals in the country. And what we're finding over and over again is that the similarities far outweigh the differences between civilian trauma and military trauma. And so what we're finding in the military cohorts makes sense for our civilian traumas, and what we're finding in our inner city, highly traumatized civilian populations also, for the most part, helps us understand better the military cohorts as well. So what about the amygdala? So again, the amygdala is an area that I'll come back to in many different ways. From a human perspective, one of the reasons why this is particularly tractable is that it is robust. And pretty much around the world, if you do a standard Wayland or otherwise fearful faces task, you're going to get activation of the amygdala in both healthy controls as well as in people with fear-related pathology. This great review by meta-analysis by Amit Etkin and Tori Wager showed that a number of fear-related disorders, PTSD, social anxiety, and specific phobia in particular, are associated with more activation of amygdala when looking at fearful faces than healthy controls. And for me, the clinical component that I am most drawn to is the idea of a panic attack. So someone who's having a panic attack will say something like, all of a sudden I was dizzy, my legs gave out on me, I couldn't catch my breath, it felt like someone was choking me, my heart was beating too fast, I was terrified I was dying, I had to get away. And of course it consists of a whole host of symptoms. And when we're training clinically, we typically associate the idea of a panic attack with panic disorder. But in fact, you have panic attacks with all of these disorders. And the primary difference is what are the cues. So with panic disorder, it seems that the panic attacks come out of the blue. That's kind of the critical component of it. And people don't know what the triggers are. Yet often, if you're interviewing a patient with panic attack, the very first ones were often at a time of high stress or severe depression or grief or whatever. But it seems that what happens is instead of associating those sort of emotional anxiety symptoms with the state they were in, for some reason they associate it with the context or with the inability to escape from it or their bodily symptoms, so that then the interoceptive cues, the next time they're feeling their stomach upset or they're sweating a little bit or they're having a harder time breathing or they're afraid they can't get out of a situation, reactivates the fear that they're going to have it again. So panic attack is almost, or panic disorder is almost like fear of fear itself, you know. Whereas simple phobia, if you're afraid of spiders and a big spider lands on you, you have all these symptoms, you just call it being scared to death. But the critical point is it's the same set of symptoms. And similarly with PTSD, when the trauma trigger is reactivated, you get those symptoms. And the reason, as a psychiatry resident, that was so exciting to me was that this was a set of symptoms that explicitly were in DSM-4, purely based on clinical observation. And yet you can directly match these up on all of the hardwired symptoms that people like Mike Davis and Mike Fanzelo and Joe Ledoux spent several decades working out as the hardwired output of amygdala activation. So we know in a rat or a human, if you activate central amygdala electrically or chemically, you get hardwired connections with downstream subcortical areas that activate all of the same symptoms, for the most part, of a panic attack. So we can really think of the fear reflex as directly tied to this set of behaviors that we call panic. And that, I think, gives us a lot of tractability in understanding, so what's upstream of that, and why are some people able to regulate this process and other people not. So before I get into all of our new data, I find it useful to sort of take this amygdala-centric view of the world and try to place a little bit of our current treatments on top of it to see where have we come and where are we going. So again, the model here is that as a final common output of these processes, the amygdala is constantly comparing the external sensory environment to internal memories, and again in this preconscious, relatively very rapid way, saying, is this something that, do I activate the fear response, do I activate the HPA stress response, or am I tolerant and can I be resilient? Do I not activate that? And I'll talk about, at the end today, about some of our work and others on understanding the fear off and what might be thought of as the resilience or the pro-appetitive networks within the amygdala that seem to be counteracting the fear-on, fear expression networks. And of course, this is being modulated by cortical areas and hippocampal areas. And so where does this fit with our monoamine models? Well, of course, serotonin is doing lots of things throughout the brain, but it's also primarily acting to activate inhibitory neurons within the amygdala, and on average, inhibits amygdala output. Certainly norepinephrine, on average, activates amygdala output. Amygdala through Koob and others' work, is known to, through CRF, project back down to the locus and have a counter-activation system here, and through ventral prefrontal cortex is able to inhibit the RAF-A, so that the whole system can be set up as one through monoaminergic circuits, and through these more complex circuits can reach equilibrium and can, in a normative state, go from a period of transient fear back to normalization. However, in states of severe anxiety and panic and PTSD, the data suggests that we have hyperactivation of the amygdala from the imaging studies, decreased activation of, and depending on which areas you talk about, this can be confusing, but decreased activation of the inhibitory cortical areas, increased activation of the excitatory cortical areas, overactivation of adrenergic systems, of CRH, HPA axis systems, and a relative dysregulation of serotonin systems, so that when these same sensory inputs come in, the system is prone to have a more stress and fear response. And our current medications, the monoaminergic and reuptake inhibitors, seem to cause these systems to come back to a homeostasis. Our benzodiazepines, of course, through acting on GABA, can lead to inhibition of the output circuits. Things like ECT presumably are causing essentially rewiring and plasticity of these systems, and similarly psychotherapy is allowing top-down new learning to modulate these systems. But where do we go from here? And can we, by understanding better the neural circuits and the molecular mechanisms of these systems, come up with novel therapeutics really based on our understanding of the circuitry? So to back up then, how do we think about PTSD in general? And of course, speaking to many experts in the audience here, but I'll just give you sort of my take on it. First, the question is, why, given a severe trauma, do only about 10% of the people develop PTSD? The vast majority recover. So there's something important that's different about those who develop PTSD who those recover. There's differential pre-existing sensitivity. Ming Swang had some of the first work on heritability of PTSD, and much of the work's come from here, and we now know that maybe 30 or 40% of the differential risk is genetic. There's also differences in how the body and the brain respond at the time of the trauma, and particularly how the trauma is incorporated in the brain and consolidated in the minutes to hours to weeks following trauma. Then once it becomes permanent, there are different cognitive and behavioral processes which seem to push one more towards the PTSD approach versus more towards the resilience and recovery approach. So it's the generalization of fear and sensitization of fear seem to be counteracting processes that lead to resilience, discriminating good from bad, being able to discriminate the fearful cues from the non-fearful cues, and extinguishing. And Pavlov defined extinction as diminished response to cues over time in the absence of the reinforcer. And we think now that the underlying therapeutic mechanism of one of our best understood therapies for PTSD and other fear-related disorders, exposure-based therapy, is basically acting on the mechanisms of extinction. So when we first, the field of psychiatry in general, starting from Ashram, Caspian, Moffitt, and others, looking at genetics of PTSD, the hope was that there would be just a few genes, and they would all make sense. There would be CERT, there would be BDNF, there'd be COMT, and several of our other favorites, and it was we'd be able to wrap it up and go home. Well, now we know that's not the case. In fact, there's many genes, if not hundreds of genes, if not thousands of genes, and the whole field is struggling with how do we identify which these genes are. Is everything that's involved in the molecular mechanisms of this by definition going to come out in genetic studies? Or are there going to be some things that are very much involved in the biology that don't come out in genetic studies, and vice versa? And so the whole field, as we know, is struggling with this. The power to do this has been led in the last 5 or 10 years through large-scale genome-wide association studies. And the best example of success so far has been the schizophrenia cohorts with the Psychiatric Genomic Consortia. And they found when there was around 10 or 20,000 samples, so the studies I'll be telling you about from Grady have 10,000 samples, which when we started we thought would be tremendously well-powered, and which we now know is quite still underpowered. And with the schizophrenia cohort, it took, once they got to around 100,000 total samples, the power looked really good. And this is a Manhattan plot. I think everybody's probably familiar with it, but just to remind you, this is the inverse log p. 10 to the minus 8 is the multiply corrected p-value level once you've corrected for 1 million different polymorphisms, and then all the polymorphisms are graphed on the x-axis here. And what this is showing now is that this is outdated. The most recent data shows something well over 100 different polymorphisms in genes in intergenic regions associated with schizophrenia. And that's now allowing for sort of a rebirth of the molecular approaches to understanding the biology of schizophrenia. What we really hope in the psychiatric, the PGC for PTSD, led by Caroline Nievergelt, Kirsten Cohn, and myself, Israeli Bazan, and a bunch of colleagues, is that when we get to the same point, and a recent R01 appears to be funded, we hope, that Caroline's taken the lead on, that plus foundational money, we hope will push the PTSD field to over 100,000 within the next few years. And again, what we're, as a field, really hoping is when we get to the same point that schizophrenia is, one, assuming that there's a similar genetic architecture at some level, we'll be able to take these genes and put them back into the circuits that we so much better understand in the area of PTSD, and that I'll be spending the rest of the time talking about. That said, that hasn't been without success. There's been 5 genome-wide significant studies published so far that have been replicated to a variety of degree. And again, the largest and most clean study so far came out of the Marine Resiliency Study. One data set that I wanted to just follow up on as far as a replication was this GWAS finding from Joel Gelartner's group at Yale. And they found a SNP that was associated with a gene called cordactin blue, which is involved in actin polymerization. And we separately, in the Grady Trauma Project cohort, were able to replicate that SNP in subjects that had a high history of childhood trauma. And again, what we're finding frequently, and I'll say more about this when I talk about FKBP5, is that if you have a history of childhood trauma, we know that one of the biggest risk factors for PTSD, for depression, for most everything in psychiatry, as you know, is childhood trauma. If you have child abuse, you're more risk for everything. Certainly from a perspective of amygdala sensitization, early childhood trauma puts one more at risk. So we found that if we look at the same SNP in this entirely separate cohort, we found the same directionality. And what was particularly cool was when we look at that SNP associated with brain structure. So this is diffusion tensor imaging, looking at fractional anisotropy and basically the strength of white matter connections. We did it throughout the brain, and the primary signal was in the uncinate fasciculus, where the SNP associated with PTSD was associated with less robust white matter integrity from amygdala to prefrontal cortex in the uncinate fasciculus. And again, this is one of the pathways that we're all most excited about in terms of top-down regulation of fear and emotional processing. So again, hopeful that although which genes are definitely going to be associated with PTSD and how they're all going to fall out is something that as a field we're really in its infancy. This and other data that I'll show you are leading us to being hopeful, at least, that eventually these genes will start to make sense in terms of the circuits they're explicitly affecting. Another recent GWAS from our group, so led by Lynn Almley with Charlie Marmer's program in the Cohen Veterans Center at NYU, and then replicated within our Grady Trauma Project, was another SNP that met genome-wide significance on chromosome 4. This is in an intergenic region, but looks like it may be a non-coding RNA. But interestingly, when we looked at this SNP's association with brain activation, so fMRI to fearful faces, so go back to that original fearful faces diagram I showed you, and do a whole brain analysis, the primary area we find significant at a whole brain level was dorsolateral prefrontal cortex and dorsomedial prefrontal cortex. So again, areas that very much have been associated with differential regulation of emotion processing and PTSD. So again, haven't progressed much beyond this so far. This just came out, but we're hopeful that this again will be evidence that some of the molecular hits will start to lay out in different components of the circuits related to PTSD. Okay, so the rest of the talk I want to break down into several points. One, I want to talk more about understanding risk and really thinking about gene by environment interactions, how we think about gene by environment interactions, and using FKBP5, which I'll tell you more about, as a model of one of the areas that has made the most progress. And again, this is the idea of how such like child abuse is interacting with genetic structure to put one at higher risk for later PTSD. do want to talk about one of the areas that is, I think, most exciting in PTSD is, again, that we know when it starts. But by knowing when it starts, that gives us, as a field, the ability to potentially intervene early right after a trauma. So if we knew the molecular mechanisms of fear consolidation, it potentially could lead to emergency department-based models where you could intervene in the immediate aftermath in the same way we have protocols for heart attacks and strokes. And there's been looking at this for quite a while in the area of noradrenergic blockade, but it hasn't panned out very well, and we have thoughts about that. But I want to show some new data in that area. And then talk a little bit about enhancing extinction of fear. And so I'll review some of our prior work in cycloserine, and then talk about some new work that we're very excited about that's unpublished in the realm of specific fear-off pathways, with the take-home message of, by understanding the neural circuitry and the molecular mechanisms of fear inhibition, can that lead to novel, targeted new therapeutics. Okay, so I mentioned briefly the Grady Trauma Project. Again, it's a civilian, all traumatized cohort. Well, not all traumatized, about 95%. These are people just who are generally coming to the medical clinics at Grady Hospital. They're not in psychiatry, but we really see it as an epidemiological sampling of impoverished inner-city Atlanta. And what we find is quite astounding. One, about 95% have had significant trauma, criterion A trauma. About 50% know somebody personally who's been murdered. About 2/3 have been attacked themselves. About 1/3 have been sexually assaulted. About 1/3 have had childhood trauma. And the story I want to tell is in many ways an old story of hypothalamic pituitary adrenal axis. So when we started, before GWAS were readily available or affordable, we wanted to do some targeted gene-wide analysis focusing on genes involved in the stress axis. And of course, just to remind us, the hypothalamic pituitary adrenal stress axis is one of the most studied and well understood processes involved in stress in general, and particularly in depression and anxiety disorders. And the amygdala projects and releases CRH into the hypothalamus. CRH is released from the hypothalamus to the pituitary, releasing ACTH, really causing cortisol release from the adrenal, which then feeds back into the whole system. And one of the oldest biological findings in PTSD that's been well replicated is the super suppression of the HPA axis. And so this is data, some of the earliest data from Yehuda and Charney and others, but it's been replicated in many ways. We've replicated in our data sets. And what we find is, if you look at blood cortisol at baseline, and then you give people dexamethasone, a cortisol analog, that night, and then look at blood cortisol again the next day, everybody will suppress. This is looking at feedback within the HPA axis system. If you have severe melancholic depression, on average without trauma, you'll have a non-suppression of the HPA axis. But most interestingly, in perspective of this talk, is if people with severe PTSD tend to super suppress. So they have similar levels, though on average maybe slightly lower levels of cortisol at baseline, but what's really profound is with suppression, they suppress much more. They have a hypersensitivity of the HPA axis feedback system. So we looked at a number of genes involved in HPA axis and asked, what is underlying this process? And this was led by Elizabeth Bender. And the take-home message was FKBP5 was one of the biggest signals. So FKBP5 is a chaperone that works with the glucocorticoid receptor, and it's part of a large complex that normally is preventing glucocorticoid receptors from translocating to the nucleus. But with sufficient cortisol being present, FKBP5 is dislocated, replaced with FKBP4, and GR translocates to the nucleus, and it does what GR is all about, which is activating transcription of a whole bunch of stress-responsive genes. But one of the most sensitive stress-responsive genes is FKBP5 itself. And so what's really nice here is now within the cell, you can see how you have a rapid intracellular feedback system that's happening at a cellular level. At the same time, you have the whole HPA axis feedback system. And the take-home message is polymorphisms within FKBP5 lead to increased sensitivity of GR binding to this, leading to more sensitive FKBP5 expression. And that methylation and polymorph and the genetic status interact together with the history of childhood trauma to regulate this super sensitive HPA feedback. So I'll walk you through the data supporting those comments. So what we first, again, were looking at were gene pathways within the HPA axis that were related to PTSD. And we didn't see much with any main effects. So we started looking at gene by environment effects. So again, as I mentioned, two of the biggest risk factors for PTSD are prior adult trauma and prior childhood trauma. And we replicated this in our group very nicely. You could see a nice linear association with multiple types of adult trauma, being associated with more PTSD after the index trauma, and similarly with the history of childhood trauma. And again, with the FKBP5 polymorphisms, we didn't see a main effect, but we didn't see an interaction of adult trauma, which I'll come back to. But we saw an interaction with childhood trauma. So that if one had 2 or more types of physical, sexual, or emotional child abuse, and one had the risk version of the FKBP5 allele, one had much more PTSD symptoms compared to the opposite allele, in which there seemed to be resilience. There seemed to be very little increase. And this looked very much like the Caspi and Moffitt finding with the CERT polymorphism. And we were able to see this with multiple polymorphisms within FKBP5 that were in LOD with each other. And again, the take-home message is one version was associated with much more child abuse-mediated adult PTSD following adult trauma than the other version. Similarly, that risk allele was associated with more significant HPA suppression, just like in the Dept suppression test. Again, one of the other biological findings that's been most replicated in PTSD is smaller hippocampal volume, and there's the field's kind of gone back and forth whether it's a trait effect, a predictor effect, or a result of stress. We can talk about that afterwards. But the basic finding, led by Doug Brimner initially, was smaller hippocampal volumes in those with PTSD compared to normal. Nagarfani in our group showed that FKBP5 risk allele associated with more PTSD was also associated with smaller hippocampal volumes, looking at microstructure analysis, as well as differential hippocampal activity, and using DTI as well, so the same idea of white matter connectivity, saw decreased robustness of the cingulum bundle between the hippocampus and the cingulum. So all consistent with the same SNP associated with more sensitive GR feedback, and more likely PTSD was also associated with differential hippocampal functioning and structural connectivity. And Amid Hariri's group showed that it was also associated with more amygdala activation. Torsten Klingel, who's working with us now and did his PhD with Elizabeth Bender, had a really nice data set on trying to put together how the polymorphisms associated with FKBP5 are interacting with childhood trauma via epigenetics to mediate this feedback. First, what he did was shown that this polymorphism that's associated with differential risk is also directly associated with glucocorticoid receptor function. So he took the piece of DNA from the FKBP5 gene that had this SNP in it that's within a glucocorticoid response element, put that upstream of luciferase as a reporter in a cell culture assay, and showed differential luciferase activation, differential transcriptional activation as a function of cortisol, with the only difference being that SNP that was associated in humans with more PTSD. They then used a technique called chromatin capture to show that the risk allele of FKBP5 was more associated with GR transcription factor binding to the polymerase start site of FKBP5 than the resilience allele. So again, the idea is this risk allele is more likely to interact with GR. It's more likely to do that and then cause three-dimensional increased transcriptional activation of the polymerase, again, leading to more sensitive feedback. And finally, they showed that if they looked at patients with a history of child abuse and this polymorphism, they saw a very interesting interaction. So if we focus on these, so this is looking at methylation of the locus and looking at the CPG sites as a function of genotype and prior childhood trauma. So if the person had no history of childhood trauma, it didn't matter. They had higher levels of methylation at these particular CPGs. If they had a history of childhood trauma and the protective allele, their levels of methylation looked the same as if they did not have trauma. But if they had prior childhood trauma and the risk allele, they had significantly less methylation at these sites, consistent with more FKBP5 expression, decreased repression of transcription by less methylation, and again, more sensitive HPA feedback. So the question that we've been left with is then, so this is a really nice model, we think, of how a polymorphism associated with gene bioenvironment interaction associated with increased PTSD could be working at a molecular level. But why is it that we consistently saw an interaction with childhood trauma but not adult trauma, or not a main effect? And it suggests that there might be critical periods during development when these brain systems are more sensitive to cortisol. And some work by Regina Sullivan has been particularly exciting in this area. And what they've done, she was at Oklahoma and now at NYU, she showed that rats undergo a very interesting period of cortisol-dependent switch in amygdala function. So they showed that if they took rats, trained them to be afraid of an odor, so they do an odor foot shock, and then bring the rats back to look at an odor choice test, they'll generally, an adult rat will avoid the odor that was paired with foot shock. That makes sense. They've learned to be afraid of it. But if they look in development, they see a very intriguing thing. Rats ages 10 days and 11 days postnatal look just like an adult. They spend less time with the paired odor shock. But a 9 or 8-day-old rat actually has the opposite paradoxical effect of spending more time over the odor that was shocked. And teleologically, they think this may make sense because early during development, pretty much their only salient stimuli are the mom stepping on them and sort of the craziness of the nest, and they want to be avoided. They want to approach that. They don't want to be running away from the nest. But this is right around the age P10, P11 when they start to wander away from the nest, they start to explore the world, and they better turn on their fear system. And so what they showed was that this switch between approach and avoidance dependent amygdala activation could be entirely replicated by putting cortisol systemically or within the amygdala. So that if you give a cort burst earlier during development, you switch the animal to an adult-like fear system more early. So what does that mean? It could mean a lot of things, but without our model, it suggests that with normative development, one ends up with a properly timed amygdala that comes up with proper dynamics between when you can be afraid and when you can recover. But with early childhood trauma, lots of data has suggested that kids in risk environments, low-income kids, kids in war-torn communities, et cetera, have higher cort early during development. That these may be set up such that you now may have an amygdala-dependent emotional circuit that turns on fear too early and becomes sensitized or primed early during development. And of course, this is only a model, but I think it's one that's testable. So now if we come back to our model of what's the amygdala doing, in a normative way, it's constantly looking at the outside world and say, do I be afraid of it or am I resilient? But through a combination of developmental cortisol that help is switching on the fear-off system, and partially being modulated by genotypes that make the amygdala differentially sensitive to cortisol, that in an early life stress situation with FKBP5 polymorphisms that are more sensitive to cortisol, one could imagine a more early developing amygdala stress system leading to higher risk later during life. So again, this raises more questions at this point than I think we have answers for, but we feel like it's a way we can start to pull together a lot of these different complexities into targetable questions. Okay, so that's one way we can think about how genetic risk, and of course we hope as the GWAS literature grows that we'll be able to understand many of the new genes in more sophisticated ways, but it's one way we can think about this interaction. What about fear consolidation? Again, one of the things that's quite exciting about this is we can take human data right after a trauma in the emergency department and look at RNA expression, we can look at genotypes, we can look at methylation, and we can compare those data to mice in which we've done fear conditioning and have a PTSD type model, but which we can look at the same thing. And in case I forget to say it, I don't really think we have good models for PTSD per se, just like one could argue for many of our psychiatric disorders. But what I think we do have is a really good mouse model of fear that I think we can do very similarly in a human. And we know, so I think we have a good mouse model of human fear conditioning. And we know that PTSD patients have dysregulated fear conditioning and dysregulated processing of this fear. So I've been talking about the amygdala so far, but we now know that the amygdala is really 14 or 15 different subnuclei. And so we're going to dive down into that a bit. And particularly, we now know that there's a whole host of different cell types within these different amygdala nuclei. The canonical pathways that the conditioned and unconditioned stimulus are paired in the lateral amygdala, and that then processing occurs in the basolateral, which then is regulated both through the centrolateral, the centromedial, by way of, in part, intercalated cells, and that the centromedial, again, activates that fear reflex we talked about earlier. And I'm going to talk about first the set of cells in the centromedial that are marked by the tachykinin 2 gene, and then I'm going to talk about a set of cells in the basolateral that are marked by Phi1 and a number of other markers. So the tachykinin 2 pathway. So this was a gene that we found by looking at genes that were differentially expressed following fear consolidation in an animal model of stress and fear. So we would, Raul Landero, who trained with Antonio Armario in Barcelona, would do a 2-hour restraint stress, then do nothing else with the animals for a week, and show that that single prior stress, sort of like I showed you that adult trauma is prior adult trauma puts one at higher risk for later PTSD. If he does a significant 2-hour strength stress, waits 2 weeks, they have a whole host of anxiety-like behaviors on open field and plus maze, decreased cognitive function. But for our purposes, they have a really nice deficit in extinction. So it's a model of stress-induced extinction deficits. Okay, so we wanted to say, what's different in the amygdala at a gene expression level in this model during fear consolidation? And again, just to remind you, the classic Ledoux, Davis, Fanzolo, Pavlovian fear conditioning, we do tones, shock pairing, the tone is activating the thalamus, the cortex together in the lateral amygdala through Hebbian plasticity. The unconditioned foot shocks occur, plasticities happening in here, such that then in the future, the tone alone is sufficient to activate the hardwired stress response. And when Raoul looked at genes that were differentially expressed following fear conditioning in this stress-induced fear model, one of the genes that rose to the top was called TAC2 or tachykinin-2. And this was particularly exciting to us because when we did in situ hybridization, we found that tachykinin-2 was specifically expressed in the centromedial division of the amygdala. And quite a bit had been worked out through Andreas Luthi and Dave Anderson and others showing that within the central lateral, there were opposing cell populations, PKC delta, which seemed to be a fear-off population, and the PKC delta negative cells, which seemed to be fear-on. And they then project to the centromedial, but there was no good marker yet of the centromedial cells, which project downstream and activate this. So the TAC2 then is a gene that's upregulated with fear conditioning in animals with prior history of stress and is specifically expressed in the centromedial. So we were quite excited about that. And what was also neat about it is that there were drugs that specifically targeted its receptor. Asanitant is a drug that targets the TAC2 receptor, also called the neurokinin B receptor, and it had been actually developed for schizophrenia decades ago. As with many of the things that were looked at in schizophrenia, as pharma was trying a whole host of different neuropeptide agonists and antagonists, it didn't have any efficacy, but it was really safe. So it's a potential drug that could be revisited with known safety. And so Raoul asked, if this gene is normally upregulated in the central amygdala during the consolidation phase of fear processing, if we block its action, can we block consolidation of fear? So he gave it right before fear conditioning, right afterwards, or several hours later. And when he then tested the animals 24 hours later, he found that if he gave it the drug right before or after, up to an hour later, he had a decrease in fear consolidation, but no effect if he gave it 4 hours later. And what's notable is, in this model, he didn't get a complete amnesic effect, but he got diminishment, and he got what appeared to be a normalization of the fear consolidation response. It didn't have any effect on fear learning itself, or on fear expression, or on conditioned stimulus response, etc. He then gave the drug directly into the central amygdala with cannula and trained the animals to be afraid, gave the asanitant, tested them the next day, and again, if he gave the asanitant directly in the central amygdala, he had a nice blockade or decrease in consolidation of fear. He then wanted to ask, genetically, could we push this in the other direction? So he made a virus that overexpressed the tachykinin and specifically expressed that within the amygdala, and sure enough, after fear, no effect on unconditioned responses or on fear acquisition, but when testing fear expression, they now show significantly more fear. So we can push the tachykinin system in both directions. And finally, he did the penultimate experiment where he combined both approaches, and he first replicated the asanitant. So in a GFP control virus, he showed a deficit in fear consolidation. Overexpressing the virus compared to the GFP increased fear consolidation, but now giving the antagonist in the same animals that are overexpressing TAC2 normalizes the fear consolidation. So it feels like we can push and pull the system as a function of this gene that we found to be differentially expressed during fear. We've previously done work, and this came from a great review by Murray, on the opioid system, which I'm not going to have time to talk about today, but the tachykinin system antagonists here, and again a number of other processes, are all starting to build up a potential set of pharmacology that we can be thinking about using in rationally designed ways to block consolidation in the immediate aftermath of trauma in the emergency department, or even potentially on the battlefield. We can talk about maybe afterwards, if people are interested, why the propranolol and the adrenergic antagonists may not be working as well as initially predicted, but I think the general idea is still a really good one. Okay, so I want to spend my last few minutes talking about extinction and enhancing the process. So after someone already has chronic PTSD, how can we use our understanding of learning, our understanding of fear inhibition to help them get better faster? So where the first work that I did involved with this was in Mike Davis's lab now about 15 years ago. And what we had done was do basic fear extinction. And just to remind you, if the condition response is freezing or startle, if you have multiple CS-US pairings, you get an increase in the condition response. But now if you repeatedly show the condition stimulus, the tone or the light or the smell, in the absence of the foot shock, you'll get a diminishment of response. And that was defined by Pavlov as extinction. And if you measure the level of fear later, you have less fear. The NMDA receptors are chock full of the amygdala. And Mike Davis's group had shown previously that learning to inhibit fear was dependent on NMDA activation. So the way they did this, Bill Falls, who's now at Vermont, took rats, trained them to be afraid, and then measured their startle to a light. So he did light shock pairing, and he showed really nice levels of fear. He then extinguished the animals normally by giving 60 lights in the absence of shock, and then tested them again, and now they're not afraid. So that's the extinction curve right there. But if he did the same experiment in the presence of an NMDA antagonist, AP5, and measured them the next day off of drug, they're just as afraid as they ever were. Showing with this and many other studies that learning to inhibit fear is an active plasticity-dependent process, and that extinction is not an erasure, which we knew from other behavior studies, but it's an active NMDA-dependent. Now we know BDNF and other plasticity mechanism-dependent process. So we wanted to know, about a decade later, could we enhance this process by enhancing extinction? And D-cycloserine was an agent that was known to act at the serine glycine site to enhance NMDA functioning, enhance calcium flow-through, and was known to be safe in humans and had been used for tuberculosis for decades before. So what we did is, instead of giving 60 lights, which would give us a floor effect, we only gave 30 lights. So we trained all the rats to be afraid, and now we gave D-cycloserine at a dose to enhance NMDA function. We had a partial extinction with 30 lights in vehicle, but we had a nearly complete extinction with 30 lights in D-cycloserine. This work has now been replicated many times. Rick Richardson's group in Australia has done some of the best work where they showed that the DCS effect is really affecting the consolidation of extinction in the hours after the extinction process, and they've worked out a number of parametrics around it. So the reason we wouldn't use DCS as opposed to other agonists was because it was safe in humans. We wanted to see, could we repurpose this drug in a specific way? And with Barbara Rothbaum, she was had done all sorts of great work in the area of CBT and exposure therapy, but also had been a pioneer in virtual reality exposure. And we thought virtual reality would be a great way to test extinction because you can control the exposure, control the aftermath, etc. So we took people with a fear of heights who would go up to 19 floors, walk out on these catwalks, hold this bar, and look down at the lobby below. Now, if you're not afraid of heights, you'll be like my kids and say, this looks like a bad video game. And that's kind of true. The graphics were 90s. They weren't all that impressive. They're much better now. But what's important is that if you're afraid of heights, you get immersed into it. And your fear system, just like my wife thinks this stick is a snake, feels like you're afraid. And they know after about 8 sessions, you can get really nice extinction of fear. And we were able to show in this study with rat, that just like in rats, instead of 8 sessions with only two sessions with 1 pill before each session, we could have people extinguish their fear much more rapidly. So 1 month and 3 months later, those who received the D-cycloserine had much less fear compared to those who had the vehicle. So since this study, this has been replicated about 10 different times and survived 3 meta-analyses. And there's been several negative studies we can talk about as well. Part of it we think is due to dosing. Part of it's because NMDA activation seems to activate whatever process is active. So if you get a little bit of activation and start to activate a reconsolidation process, NMDA activation may strengthen the reconsolidation, whereas if you have a full extinction, it strengthens the extinction process. And we can talk about that more too. But the take-home message is that, at least proof of concept-wise, with exposure-based psychotherapy for social anxiety, for OCD with exposure and response prevention, for PTSD in a number of studies, and panic, it all seems to enhance the rate of learning to inhibit fear across these studies. And I don't think D-cycloserine is going to be the be-all and end-all for many reasons, but it was really exciting to us and to me as a model in which we could take basic learning principles of neuroplasticity and apply them to how we think about psychotherapy enhancing learning processes, and give us maybe a whole new approach to thinking about making new treatments that more specifically target recovery. So in my last few minutes, I want to talk about where we are now with those kind of approaches, trying to identify new pathways and new drug targets and molecular targets based on a more sophisticated understanding of fear inhibition than we had a decade ago. So again, with things like D-cycloserine, NMDA agonists, BDNF agonists, even our monoamines, these are very broad-acting drugs that are really affecting the whole brain and are going to always have off-target effects of plasticity in addition to the specific effects. So one way we can target it in a specific way is timing. But what if we could also target plasticity as a function of molecular specificity? So one of the groundbreaking findings in the last decade about how the amygdala processes emotion came from Andreas Luthi in Basel and Cyril Harry. And what they did was they recorded blindly from rodent amygdalas, and they did it during fear conditioning and fear extinction. And they showed that about 30% of the cells didn't seem to respond to their tones no matter what. Another 30% they called fear neurons. Those did not respond to the tone before tone shock pairing, but after tone shock pairing, they responded to the tone as if they were encoding the tone in a fear-dependent way. But once the tone had been extinguished, they didn't respond to the tone anymore. So it seemed to only hold the tone memory, if you will, or engram, while the tone represented the fear response. Another set of cells that seemed to be intermixed with these did not respond to the tone initially and did not respond to the tone after fear conditioning, but now did respond to the tone after the tone was extinguished, such that one set of cells responded after fear conditioning and diminished its firing with extinction, and another set of cells only started firing to the tone when those cells seemed to be holding the fear extinction memory process. So in addition to what we now know about a lot of top-down regulation, say infralimbic cortex projection to the BLA and to the encirculated cells, there's also these set of cells within the basolateral which also are holding this fear inhibition memory. So it seems like there are two opposing hypotheses for how these cells could come to be about. One could be that all pyramidal cells within the basolateral, these are all pyramidal excitatory cells, that they could all be stochastically determinant to either become a fear-on cell or a fear-off cell, just depending on their firing history and their history of the neighboring cells and the pyroalbumin inhibitory cells around them. An opposing possibility is that these are genetically determined cells that are biologically different cohorts of cells. And if the latter were true, then that would suggest if we could know who these extinction cells are, or alternatively, if you know who the fear-on cells are, you could then push and pull them in opposite ways based on what receptor cohorts they express and what genes they express, so that you could start to have a molecularly targeted pharmacology on these specific microcircuits. And at least one piece of data that suggests that these are biologically distinct cells is that we know that there are different cells in the basolateral that project to the central amygdala and the BNST, our fear-expressing and anxiety-expressing pathways, and separate BLA pyramidal neurons that express to the nucleus accumbens, which we think may be associated with both addiction and appetitive behavior, but also may be associated with inhibiting the fear-on behavior. So as we were starting to get into the world of optogenetics about 5 years ago, we were doing a lot of virus work, but the viruses were slow and cumbersome and caused a variety of problems. So in parallel with that, we found this population that Josh Sainz had made that was available from Jackson Labs, in which a Thy1 marker, which is a specific receptor that's known to be in lymphocytes, but also in a few parts of the brain, including a subset of pyramidal neurons in the cortex and in the amygdala. And what's interesting about this Thy1 marker is it's not expressed anywhere in the piriform cortex, temporal lobe, except for the basolateral amygdala. And it's not expressed in all the cells. It's only expressed in maybe a third to a half of the CAM kinase positive cells. So we have this available channel rhodopsin line. And again, with channel rhodopsin, you can activate via optic fiber. You can send down a laser light, which then causes the cells to fire. And so we could ask, electrophysiologically, what are these cells doing? And behaviorally, how do these cells affect fear? So electrophysiologically, by themselves, they cause the BLA to fire. But what was interesting, this is all work with Don Rainey's group and Aaron Jasnow, that if we stimulated the lateral amygdala in a slice and then measured the central amygdala output, we would normally get firing of a subset of cells with this flow-through, again, sort of the canonical amygdala flow-through. But if we did a subset of cells for which we activated the central amygdala via LA firing, if we now activated with optogenetic light the BLA intermediate cells, it completely blocked that flow through. It looked like it was inhibiting the amygdala flow through. Okay, so what does that mean for behavior? So Aaron showed that if he took mice now that are running around, and at the time of the foot shock in red, he also activated the basolateral amygdala, and then he did all sorts of controls, either genetic control or didn't turn the light on. We had no effect on acquisition of fear, but the animals that had laser activation of the Thy1 cells showed essentially no fear consolidation. It was how you completely blocked the fear learning process. So we thought, well, maybe that's because we're turning on a fear-off population, or maybe we're just scrambling the amygdala. Well, if we know from Musammal experiments and a whole other things that if we scramble the amygdala, we'll block fear consolidation, but we'll also block fear extinction, just like I showed you with Mike Davis's AP5 experiments. So our hypothesis was if we did the same experiment during extinction and we blocked extinction, then it was probably non-specific activation that was just scrambling the signal. But if we enhanced extinction, that would be consistent with pushing fear down in both processes. So Aaron trained animals to be afraid with the foot shock, then brought them back the next day, and at the time that they would have received the shock after the tone, they now got the laser activation of Thy1. And sure enough, they had a normative extinction process, but when we brought them back the next day, animals that had been laser activation of the Thy1 BLA cells at the time of extinction again showed no fear. So those data were consistent with turning off or activating a fear-off population. Since that work, Kenneth McCullough in our lab has done a fair amount more work to characterize these cells. First, he's shown with FOS activation that the YFP positive, so the Thy1 marked cells, show more FOS activation during fear extinction compared to fear acquisition or fear expression. And the YFP negative cells, so the neighboring pyramidal neurons, show more activation to fear conditioning compared to fear extinction. He also did the opposing experiment. So instead of channel rhodopsin to activate the cells, he got a Thy1 line expressing halo rhodopsin to inhibit the cells. And the prediction is if we inhibit the fear-off population, we would have more fear. And sure enough, that's what he saw. He did acquisition, and then with stimulation of fear expression, he saw much more fear expression when the halo rhodopsin was activated, so we were inhibiting the fear-off population. So he's also pushed and pulled this with dreads as well. So it looks like everything we can do to these cells is is consistent with the hypothesis there's a fear inhibition or fear off population. A very interesting finding came out of Memphis, Scott Helton and Tony Reiner's work, in which they have a model of TBI, which I know is of great interest to a lot of people here. And this is a closed-head TBI blast model. And what they see is an extinction deficit with TBI. So if they do a closed-head TBI blast and they bring the mice back and they fear condition them, they then see that the mice with the TBI exposure have fear for longer and have an extinction deficit. After seeing our paper, they got the Thy1 GFP mice from Jackson, and they showed, intriguingly enough, that after the TBI blast, they had a significant dropout of the Thy1 cells within the amygdala. So this remains to be replicated, and we don't really have a good model of understanding it yet, though, as Dr. Swung and Caroline and Duane and others know there's a lot of excitement about inflammatory processes with both TBI and PTSD. So you could imagine that maybe this fear-off cell population is for some reason more sensitive to inflammatory processes. Okay, so the final experiment was Kenneth then wanted to know, what do we know about these Thy1 cells? Can we understand them more? Thy1's a marker that we don't really understand what it's doing. If these are truly genetically different, we should be able to identify other genes. So he did cell sorting of dissociated fluorescent cells and then did RNA sequencing to say, can we identify other genes that are specifically expressed within this basolateral population? And lo and behold, it worked. He was able to find a whole host of different genes that seemed to specifically be expressed in the BLA, and that seemed to be expressed in this one pyramidal population compared to another one. So to then test our hypothesis, he said, can we find a gene that's expressed specifically in these putative fear-off cells in a receptor that we have drugs available to target? And then can we give that drug, and will it affect the pathway the way we think it would? One of the more specific and highly expressed genes was the neurotensin receptor, and specifically the NTSR2. And neurotensin's had a long and interesting history in psychiatry, though we still don't really know what it's doing with all these circuits. But interestingly, NTSR2 is expressed preferentially within the BLA Thy1 population. And I'd never thought much about neurotensin 2, much less knew anything about how to target it. But it turns out from NIH you can get a host of agonists and antagonists, and beta-lactotensin is a specific agonist of the NTSR2 GS-coupled receptor. So he gave this drug with fear conditioning and then tested them the next before fear expression. And again, and he showed, much to our surprise, a profound diminishment of fear expression based on activating the NTSR2 receptor. So to us, this was very exciting because we had never thought about NTSR2 before, but based on identifying it in a specific subset of cells that based on our physiology were predicted to be fear-off cells, we were able to predict and then show that activating this receptor would essentially turn off the fear process. So there's a lot of work to go and a lot of genes and follow-up to do, and we'd really like to now start to look at these genes and compare them with the human genes associated with PTSD, predicting that a subset of human genes associated with PTSD are going to be genes that are specifically differentially expressed in the fear-on and fear-off circuitry, et cetera. But we think the future is bright. So I walk you through how we think about fear broadly, how current genetics and GWAS approaches are giving us new targets, and how we can start to take these gene targets and better understand how they're intersecting within a really well-defined circuitry of fear modulation. And again, this isn't all of PTSD. Most of these models aren't necessarily going to help us very much with things like shame and guilt and other components. But we hope that doing this sort of bottom-up, reductionistic enough but still very translationally informed approach will help us with certain components of PTSD, and by making progress there, it can make progress in other ways as well. The work that I talked about, I've really just been the cheerleader, and it's many, many great colleagues and students and postdocs. The human work has been in large part through the Grady Trial Project, led by Beck Bradley, Tanya Ivanovich, and myself with Elizabeth Bender and her colleagues in doing a lot of the genetics. And the animal work I showed worked particularly from Raul Landero and Aaron Jasnow, who's now at Kent State. Thanks so much for your attention. Yes, I guess we have time for questions. So, okay. Thanks, Kerry.
Hang on, where?
Oh, we have to do it officially.
Can we enhance learning by faculty in a different
Yes,
way?
everyone has to take D-cycloserine before the next grand round. Okay.
My understanding is that progesterone can enhance fear extinction, and that based on the work of Catherine VA, oxytocin blocks the effects of CRF. Can you address the hormonal modulation fear response?
Yeah, I mean, I would agree with both of those, and certainly progesterone. We know that males and females modulate many of these systems in different ways, and I didn't talk at all about our PACAP data, but we've also found data that's associated with PTSD in females but not males, and we think that's in part due to estrogen modulation of the PACAP transcription. So I think there are going to be many of these stories, whether everything from NPY as an enhancer of extinction to oxytocin, and then progesterone and estrogen in specific but non-linear and non-simple ways, modulating differently extinction, generalization, discrimination. So I don't think there's any simple answer there other than yes, they're going to be important, and I think we have a long way to go, but hopefully as a field we're doing a better job of breaking down the question we're asking, because I think the take-home is this drug is good for PTSD or this drug is bad for PTSD is too simplistic of a question that will probably lead us to very confusing results. But if we can say this drug is good for extinction modulation of contexts and this drug is good for diminishing sensitization of specific kinds of fear cues, we might be at an intermediate phenotype level that some of these hormonal results will start to make a lot more sense.
Thank you, Kerry. It's a very comprehensive review of everything under the sun. And I mentioned briefly to you the PTSD patient, essentially the marine resilience sample before they are sending to Afghanistan or Iraq. We collect the blood sample. And comparing those who develop a PTSD after they coming back and who remain to be non-PTSD.
Right.
And what we found was that before going there, those who develop a PTSD actually had the gene expression regulation in the RNA system, the system involving inflammation, immunological aspects of it. And we don't have the data for the cheerful abuse of these patients. From your study, we need to look into that.
Yes.
So from your perspective, we are now looking into those systems of what neurobiology involved.
Yes.
But if inflammation and the immunological system is involved, probably before going to Afghanistan, we should use aspirin.
That's right.
To calm down their inflammatory system. Probably could prevent, but this is just the hypothesis.
Yeah. Thank you,
so
Professor Swan.
what's your take on this?
Yeah, thanks, Professor Swan, for that and for all that you've done over the decades in this area. I mean, I think inflammation's clearly playing a terrifically important role across so many psychiatric disorders that we're only starting to understand, and certainly in the stress disorders as well. You know, and I think how the glucocorticoid system as an endogenous way of trying to dampen down inflammation, you know, how is that yin and yang out of dysregulated as well. And then there's so much we don't know about how inflammation is, even though there's beginning to be a lot of data on inflammation and risk, we don't know how it's interacting with any of these circuits. We don't know, is it activating through microglia or astroglia and regulating local circuits that way, or if it's directly targeting different sets of neurons. So I think that's going to be really an exciting work for the next few years, is how do these systemic inflammatory signals interact with these specific circuits.
Of course, it's a trade-off. They'll be less anxious, but bleed more.
That's right. You may die, but die happy, I guess.
Thanks for the great talk. I was very interested in finding the characterization of some of the subnuclei in the amygdala, where you found this new marker for a subpopulation. I think it was the
TAC2 centromedial population
expression. I wonder if you had the chance to perform a developmental study to see whether that expression early on, when fear conditioning is kind of affecting the system to learn what the important cues are about fear, is actually showing an increased expression of that gene or any other gene that sort of primes the system so that later on the same cues sort of show a sensitized response to fear conditions.
Yeah, no, we haven't done it. It's a beautiful idea, and I'm going to steal your idea if that's all right. We're collaborating with Regina Sullivan and a number of other things, and I didn't talk at all about any of our olfactory intergenerational work, but she has some really interesting findings in that realm, and it would certainly be very interesting to take a lot of what's now being understood at a very molecular subcellular level and put it back into that really nice model. But no,
yeah, and if a Cree line exists, it would be nice to find other genes that specifically in those identified cells are changing receptor for certain.
Yeah, and there is a TAC2 Cree line, and so we've been able to show that if we optogenetically drive the TAC2 Cree line, we can get predicted effects in terms of furthering fear expression, et cetera.
Wonderful.
So great.
Thank you.
Thanks. There's arm wrestling to see who goes.
Tell me a little bit more about the cycloserine research with PTSD patients. Specifically, I'm thinking about when we do exposures, so little of the exposure happens in the office.
Yeah.
And outside of the office, it's in vivo exposure,
Yep.
it's listening to the memory that they told to the therapist in session every day. Where would that be heading in terms of where should we be thinking about hitting that might be most helpful?
Right. So, several things, we've learned a lot about, as with most experiments, you learn more about what doesn't work than what does work, right? So with D-cycloserine, it was known. So an interesting caveat of DCS is that well before we started using it, it had been used in a number of Alzheimer's trials as well as schizophrenia trials. And because this knowledge that it was an NMDA partial agonist was quite clear in the 80s. But most of the human Alzheimer's and schizophrenia trials were quite negative, and so it was pretty much being pushed aside. When we got back into this, that was our first thought, well, this didn't work. But let's look at it a little bit more deeply. Well, it turns out that the very early data showing its cognitive enhancement effect showed that it worked if you gave it acutely, but if you gave it several days in a row, you totally ablated the effect. And that was also known that at the cellular level, you get tachyphylaxis of the NMDA receptor. And we're seeing the same thing with BDNF activation of tract B. And so my current pet hypothesis is that anything we're doing modulating plasticity directly probably has lots of compensatory effects that means you can't do it chronically. And so we know with DCS, if you give it once a week, it seems to work, tied to a specific learning event. But if you give it every day, you lose the effect entirely. So what does that mean in terms of how we do therapy? So I don't think it's something that you could take every day in those. And what we also know, again, because some of the negative data with DCS looks like the way it's being explained is in animal studies, it's been shown that if you give DCS before a single reactivation, so you're basically activating reconsolidation, it actually strengthens reconsolidation. If you give it before a full extinction, you diminish fear and you enhance extinction. So how do we have better markers in humans of whether our therapy has reactivated the memory and then they dissociated or whatever and they don't really extinguish versus do we get a good full extinction? So I think we're going to need to push more on our understanding of therapy to be able to better understand how to combine cognitive enhancers with it.
Yes.
Thanks very much. I'm wondering if you can comment on to what extent you think opiates and benzodiazepines may interfere or maybe even enhance extinction. I'm thinking in particular of some of our clinical populations, trauma populations who get liberal doses of opiates and benzos and how that might impact their extinction and consolidation even.
Yeah, so it's a bit of a messy literature. Our own data with Pavlov suggests that benzodiazepines at the time of extinction are associated with less robust extinction consolidation. And kind of the way of understanding that historically, I think, in the therapy world is that you're essentially, it's almost like a blocking effect. If you diminish the anxiety so you're not able to have sufficient anxiety to then extinguish it, you're not doing that. And or it's a state-dependent effect in which you're basically extinguishing during the state of positive benzodiazepine activation, but then without benzodiazepine activation, that learning effect wasn't encoded. Opiates, I think there's less knowledge about how that specifically interacts with extinction. I mean, obviously, with both of these cases, if we had a drug that was both anxiolytic and pro-cognitive, that would be, the perfect thing. I don't know if those exist yet. They seem to go kind of in opposite effects the way we usually understand it. What I didn't talk about was some of our opiate work in the immediate consolidation process after the initial fear warning, and that looks like opiates may diminish consolidation, but again, the opiate system is very complex, and whether it's the nociceptin pathway more so than the mu and kappa and delta, et cetera, I think has to be worked out. But I don't know of much work so far with the opiates in extinction.
So over lunch, I asked Kerry about, our favorite drug here, marijuana. Since we're talking about drugs, can you comment on that?
Yeah, so, I mean, we could have lots of fun comments, but I'll try to keep it to the data. One of the more profound apparent mediators of extinction was shown in the 80s to be the endogenous cannabinoid system. And so Lutz and Giovanni Marscano showed that if they took a cannabinoid 1 receptor knockout, the mice had a deficit in extinction consolidation. And then a number of studies showed that, if you gave a Fa inhibitor, so you basically enhanced endogenous cannabinoids, you would get a faster extinction. And if you blocked cannabinoid functioning with the SR compound that's a cannabinoid antagonist, you would block extinction. So all that suggests that marijuana is a good thing. The thing is, but there's always a but. We and others also showed, however, that although enhancing endogenous cannabinoids enhance extinction, if you gave a THC or a Win compound, so the more lipophilic direct agonists, that you actually did not have the enhancement of extinction effect. And again, I think it's back to the tachyphylaxis issue. I think the cannabinoid direct agonists are so lipophilic that you're more likely to downregulate the receptors. And you tend to see quite a different fear extinction profile if you use a reuptake blocker of endogenous or a Fa blocker of endogenous versus if you give a direct agonist. So I think it's mixed. Certainly many of our patients, PTSD patients and veterans, like marijuana. They say it's self-medication, but I don't really know if that's more in the, it makes you feel good while you're high, but whether it's really doing anything long-term is not so clear.
Good, thanks. Could I ask one other question? So you had mentioned the interaction of genetics, childhood trauma, and so forth, Well, one of the other outcomes of childhood trauma, of course, is psychopathy, ASPD, folks who traditionally are thought to be very hypo-arousable. Have there been any studies of these two, if you will, extreme populations? Because most things compare, quote, normal people to PTSD, but what about these others?
I mean, I don't know if they've been done in the child abuse neurobiology way. Certainly our experience and our data sets are that our highly traumatized women have PTSD and depression, and our highly traumatized men are in jail. So, you know, we do know with PTSD that you can get opposing effects, right? You can have your very hyper-aroused, hyper-emotional patient with PTSD, and then you can have the very numb and shut down patient with PTSD. And I certainly believe that, you know, it may be that they're more similar than they are different, and it's a matter of differential emotional responding. I'm actually giving a couple talks this next year to court systems and sort of meetings of judiciaries doing in who do child legal advocacy and this sort of work to talk about childhood trauma again and this concern. But I don't think we have nearly as much data as would be nice to have.
Good. I think we have. Did you have another question, Barbara, or comment? Or well,
the childhood trauma, of course, is really interesting, and your TAC2 data are just kind of exciting. And they kind of fit very nicely in some of the last ACNP and biological psychiatry conferences. There have been nice lectures of the change in the neural circuitry as it relates to childhood trauma. And so I'm wondering how these are interacting with each other. Yeah, have any thoughts about this?
Yeah, I mean, I think sort of like the prior question, I think it'd be great to look at TAC2, you know, both with normative development as well with childhood trauma. I mean, my prediction would be most of these pathways that are associated with amygdala fear responding we're going to see some marker of sensitization following childhood trauma. I mean, I think the question, kind of the next question beyond childhood trauma is bad, is why do some people become more resilient with some level of childhood trauma and other people become more risk? You know, so it's sort of taking this big question and then making it even more specific to childhood trauma. And, you know, one of those I think is socially, we know having a positive object, and there's a lot of things that are protective. So is it sort of the total additive amount of protective stuff versus negative amount?
okay. Final comment then.
When would you predict to be the best time to give prazosin, and is there any to maximize its therapeutic benefit? Is there any prophylactic?
I mean, the prazosin literature has certainly been very interesting, and I know the large-scale study was more mixed, but certainly there's been a lot of smaller studies suggesting works very well, and I think many of us who treat patients with PTSD who have severe nightmares and hyperarousal have a really interesting response to prazosin. Maybe I'll end on what I think is a really fun clinical anecdote. So I had a patient who was a lawyer, and she theoretically should have been very high functioning, but she'd been raped previously and had a secondary trauma in law school, and essentially had very severe PTSD, couldn't get out of her house, was not able to work, and had a lot of interpersonal trauma that was all very complex. She'd had therapy, she did a number of things, had her on a number of drugs, was not making much progress. So I tried a lot. Nightmares were one of her bigger issues. So the issue with prazosin being an alpha blocker is, you know, you get hypotension. So most people, you know, for blood pressure, a few milligrams of prazosin is often enough. I had to titrate her up to about 15 milligrams. This is a very small woman. Before she never got hypotensive, but it took about 15 milligrams initially for her nightmares to get better. Then she was sleeping better. She was doing therapy. We even tried desilocybin. Over the course of several months, every time I saw her, she would be getting better, but then she'd say, you know, I'm getting a little dizzy in the morning. And we pull it down a little bit. And so basically over several months, as she got better in many ways, she was also now sensitive to the hypotensive effect of prazosin at 1 milligram. And then we took her off. So you could almost watch her adrenergic system renormalize and then be sensitive to the side effects of prazosin. So I don't know what to do with that, but I think it's a really cool story. And I don't think there's a whole lot of data other than sort of using it for nightmares right now. Thanks so much, everybody. Great question.
Thank you.
Oh, I get a present?
So you're already an honorary member of our department. We're now making it official by giving you the UC San Diego School of Medicine Department of Psychiatry baseball hat.
Excellent.
Do not dare go to a Boston Red Sox.